DNA replicates. Okay? What else does it do? This is what you just learned last lesson. What else does it do? Good, right? So there are 2 steps to make a protein. What were those 2 steps again? Good. Transcription, translation. Good. Abhishek, what does transcription mean in layperson terms?
When the messenger or the RNA...
Take that out of your head. What does transcription mean? If you were working on a video editing software and you have to transcribe the audio, what does that mean? One word.
It creates a complementary string.
Not in video editing, right? I'm asking generally, what does transcription mean? Copy. That's it, right? So when you guys are learning biology, the thing is, don't get over detailed in terms of the knowledge straight away. Get that overall point and then start delving into the detail. So the gist here in transcription is we need to make a copy of the DNA, right? And it's because DNA is important. Where does DNA sit, actually? In the nucleus. For a eukaryotic cell, right? It can't leave the nucleus. Outside the nucleus, there are enzymes that will destroy almost immediately. Okay? So we need to make a copy of that DNA that can go to the organelle that makes proteins. What is the organelle that makes proteins? That's the R. Exactly, right? So the ribosome is the organelle that makes the protein. Where does it normally sit, Tashbia? In the endoplasmic reticulum, making it rough, right? Very good. Now, what does translation mean? If I asked you to translate something, what does that mean to you? Google Translate. You've all used it, right? How do you say hello in another language? One language to another. That is what translate means, right? So what happens is, we go from the language of bases, a 4-letter language, A, T, G, and C, to another language that has how many letters? Or how many variations? 20, that's right. What is he talking about when he says 20 variations? Amino acids, right? So we go from the language of bases, 4 bases, to the language of 20 amino acids. Good, and transcription is copy. Good, now I'm going to start going into the more detailed parts of these 2 points. And before I do, all of these processes, in the end, what do they produce? Transcription and translation. What does it make? What's the difference, Georgina?
A polypeptide is a bunch of amino acids and a protein is a bunch of polypeptides.
Okay, so a polypeptide is a chain of amino acids. Hopefully everyone got that. Hi, come in. Chain of amino acids. Good. So the definition of a protein is 1 or more polypeptides. Good. We'll talk about that soon. Now, why is the most important molecule in a cell only making proteins? And this links to a latter part of your module. What do proteins do? Abhishek? Why is DNA only making protein? You're composed of fats, carbohydrates, acids, bases. Why is that?
Proteins carry out various...
Good, so you guys understand it's diverse functions. Now let's go into what are the key functions of proteins. How would you break it down? This is how to think, right? If your brain goes straight into the nitty-gritty details, you're not thinking right for bio. One thing I want to teach you all, this is generally learning, how to learn, is this word here. You should be teaching it more at school. This is an acronym, and it's called MEISI, which means mutually exclusive and collectively exhaustive. Does anyone know what that means? What does it mean to be mutually exclusive?
Independent.
That's it, right? It's independent. You always learned about independent assortment. Mutually exclusive means they're separate to one another. They don't have any overlap. You guys seen the Venn diagrams? It's when 2 topics have no overlap at all. What does collectively exhaustive mean? When you add everything up, that equals the total, meaning you covered everything. So whenever you're trying to answer a question, in biology, chemistry, in science in general, you want to have an answer that is broken down into systematic categories that are separate to one another and collectively cover everything. That is what I'm talking about here. Make sense? So when I ask you for the function of proteins, what are 2 categories you can put in? 2 boxes that are separate to one another. Very good, right? That's how you think in terms of biology. Very good. So we have structural and functional. Pranav, what is an example of each?
So structure.
Good. Lookman, do you have a question? I can see your hands.
Yeah, no, I wanted to answer what you were asking.
Perfect. Okay, I guess Lookman, take it away. Give me an example of a structural, then a functional.
So a structure would be if doesn't protein form the cellular cytoskeleton in the cell? Yep.
Yeah.
And then functional would be antibodies and enzymes or something like that.
Okay. Okay, we're not going to cover everything for now, but he's mentioned some very important points. I guess all I'll say is the structural one you should know, which is very important, is muscle tissue. Muscle tissue is made up of protein. And the one functional I would remember right now is enzymes. We will add upon all of this later. Antibodies, cytoskeleton, we'll talk about that later. But I just wanted you all to get the gist. Now, going into a bit more detail, let's zoom into the first step. Transcription. We'll start with Kashmir. I want you to give me a one-line description of transcription. I'm going to ask each and every one of you until I get an answer I'm happy with. Now we'll cut you off. Keep on going. Take it away. Okay, sure. I want you all to try answering in an exam format. Last lesson, what you learned is the theory. You learned what it's all about. How to write it in an exam is a whole different game. And I think you might have realized that from year 11, right? The student that knows the most isn't always the student that does the best. Okay? Georgina, give it a go. What's an exam answer? And all of you should be thinking of it now while we are going across.
It's when DNA made into a compound and mRNA makes the DNA.
Good. Have you learnt? I'll come back to you, don't worry. Back table. Ria?
Process in which RNA polymerase complementary mRNA.
Good. Good. I like that. Did you see the two pieces of info she had? As a marker, that's what I'm looking for. I don't care how much depth you have. If you have the 2 points, you get the 2 marks. It's that easy, right? That's how they mark you. So RNA polymerase is one that I'm looking for, especially for trials. The second is a complementary strand of mRNA. What does mRNA stand for? Very good, right? So we're producing a single-stranded copy of DNA going to leave the nucleus. That's the gist. That's all you need to know. You don't need to know too much about antisense or sense. Can anyone tell me what that is, though? Good to know, but you shouldn't have it in your answer. It's too much. Yeah, if you look here, I kind of labeled it for you, right? The sense strand makes sense. It's the actual strand that has the base letters that tell you the protein that you're going to make. The antisense is just the complementary strand. So what RNA polymerase does, it makes complementary bases to the antisense strand. Do you all see that? And by doing that, it has the exact same letter sequence as the sense strand. Have a look at this. The sense strand is TACT. By being complementary to the antisense strand, which is ATGA, the RNA transcript is the exact same, minus 1 base difference. Uracil, right? So that's the unique feature of mRNA. Good. Abhishek, what are 2 other differences between DNA and mRNA? Or RNA in general?
mRNA leaves the nucleus.
With actual structure. What are good, single-stranded, has a uracil. There's one more that I'm looking for. Yeah, it has an extra oxygen. The sugar, remember it's called ribonucleic acid for a reason, not deoxyribonucleic acid. The sugar, the ribose sugar, has an extra oxygen atom. Very good. That's all you need to know about transcription. Now, we spoke a little bit about post-transcriptional modification. So this happens in the nucleus before the RNA moves out to the ribosome. This is only examinable in trials. Okay? In HSC, they'll give you this knowledge all over again. You have it in a little extractor table. What happens here? Can anyone tell me? I will get Harshil. Do you mind telling us a little bit about post-transcriptional modification? What goes on? Okay, we'll move on to Lookman.
splicing happens where introns are removed and exons are joined together.
Alicia, what do you understand about introns and exons? Nisha?
Introns are the ones that are not expressed, and extrons are the ones that are expressed.
Okay, what does expressed mean, Georgina? Do you want to give it a
When they leave the nucleus, it's like, can you explain also what I believe?
Yeah, kind of, but think about it. What happens when you leave the nucleus? You get made into a protein. Expression of a gene is when it's made into a protein. When you say a gene is expressed, that gene was made into a protein. That's what gene expression means, right? So exons are the actual part of the gene that make protein. These introns are regions of DNA, letters, within a gene that are kind of just nonsense. They don't make any sense, and they should not be part of the protein. Does that make sense? So a spliceosome, the way I think of it, is like Fruit Ninja. You all played Fruit Ninja, right, man? How old were you all when that came out? Yeah, like I think I was playing that in high school, which is, yeah. But Fruit Ninja, you start cutting all the fruit in random directions, right? It's the same principle with the spliceosome. It'll start cutting all the introns out of the mRNA, so it's only made up of exons. And I remember exons has an E for expressed. That's how you remember things. Okay? Very good. Now, what else did I say about the cytoplasm? Dangerous. The dangerous place, right? It's kind of like the US. There's a lot of guns, a lot of weapons, a lot of enzymes, right? So you need to protect this RNA because you're trying to make a protein. So you put armor. What armor do we use, Kashfia? Done. That's it. That's all you need to know. We'll move on. Okay? So the poly A tail is just adenosine, right? So it's a variant of the base. And the 5' cap it's called guanosine. Don't need to know what that is. It's a variant of guanine. It's chemically slightly different. Good. Now, we're going to talk about translation, which is the key thing I hope you all understood from last lesson. I'm going to ask you all. We'll start with Varun. What happens in translation?
So the RNA strand is entered through, like it goes through the ribosome and the anti-cut part, the top part of the ribosome, the big part, it translates the RNA strand microstructure called codons and it matches with the RNA molecule which contains the.
Good. I like that. Now, how many of you all can close your eyes and see the process happening? Be honest. Because you can't understand it from these words. You can't understand a movie. If I gave you James Bond or Spider-Man 3, the newest one, and you just saw the transcript of the movie, would you really understand what's going on? Same process here, right? You can't learn bio just by reading. So I want you all to write this down. There's a video called Crash Course Biology, Polypeptide Synthesis. I remember when I was in your shoes, this made no sense to me until I watched that video. And then it all just clicked. Okay? So go home, watch the video, annotate it, and you'll feel like you reached a new epiphany with polypeptide synthesis. It'll make much more sense. Okay? In the meantime, we're not going to watch YouTube videos together. You can do that yourself or at school. I'm going to go through the key points you need to understand, especially for an exam setting. Now, the first thing I want to make very clear is what moves against what. You all understand that the ribosome is kind of like, you've all eaten at Subway, right? It's kind of like the Subway bread. An actual filling is the mRNA. It sits in it. Now, what moves against what? Is the mRNA moving along the ribosome, or is the ribosome moving along the mRNA? Very good, right? So that's the first thing to understand. The ribosome is reading the mRNA. The mRNA just sits there, wedged inside the ribosome. And I'll get images up so you truly understand this. So this is a ribosome, the green, and the pink is the mRNA. It's going to be wedged inside, as you can see, and the ribosome will chug along the mRNA reading it. Now, just like how we read words, right? If I say the letter T, does that have any meaning for you? Not really. But if I say the letter 2, you immediately think of a quantity, right? In the same way, the ribosome reads in words. And in its words, it's in combos of 3. We have a name for every set of 3 bases. Varun mentioned this word starting with C. Raya, what is that word? A set of 3 bases, what is that equal to? Do 3 bases. Okay, we will ask. Good. 3 bases results in 1 codon. Now, why does it read in 3 bases? What does a codon actually help the ribosome do? Pardon? Yes, that's true, but I'm asking why not 4, why not 5? Each amino acid has 3 bases. Does an amino acid have bases in it? Good. Okay. So what happens is every set of 3 bases results in the ribosome calling forth 1 specific amino acid. That's like a currency conversion. Every 3 bases you have, ribosome can swap it with an amino acid. Okay, that's what's going on here. Okay, so let's say the first set of 3 bases will give me a random sequence. Georgina, if you say T, I will not be happy. Quickly. CAG. Right. Okay. So CAG results in a specific amino acid. Or it could be 2 or 3 amino acids, different ones, but usually it will result in 1 specific amino acid coming to the site. Okay. So let's say CAG codes for arginine. That is the amino acid. Now, remember, we're sitting in a cell right now. Imagine you're in the cytoplasm. Where is the amino acid? Does the cell make it from scratch? Where does the amino acid come from? Nucleus? Not quite. Pardon? Yes, but what? How did it get to the cytoplasm? But where did the tRNA get it from? You guys could be asking us questions, right?
I asked that last week. They said the tutor was free floating, just like the nucleus.
Yeah, it is free floating, but again, how did it get there? What do you all eat? What did you have for breakfast? If you look at the nutritional value, if you actually got like food poured out, let's say protein and an amount, right? You break down the protein into amino acids and you store that in the cytoplasm. So it's present in the cytoplasm, and that's what your cell is picking up right now. Does that make sense? Okay, good. So arginine is sitting there because you might have had a McDonald's burger lunch and had protein, which had the amino acid arginine. Make sense? Good. So now my question is, how is this ribosome going to get arginine and link it up with the rest of the chain? Because what's going to happen now is the ribosome will now move to the second position. It might read a, let's say, a CUG, right? And let's say that codes for lysine. My question to you is, how is the ribosome going to get the arginine, get the lysine, and link it up to form a polypeptide chain? Any ideas? Good. So we mentioned the word tRNA. What does T stand for, Tashbia? Transfer, right? It's because it's transferring something. What is it transferring, Alicia? Yeah, the tRNA. So this is a ribosome. This is the mRNA. So let's say the ribosome just read CAG. It wants to get an arginine. How does it get the arginine? It sends signals to a tRNA molecule. Okay? So what's going to happen now is a specific tRNA molecule come along and it will bind to the first codon. Okay? Now, what is this codon? It's CAG. Everyone with me? What are the components of a tRNA molecule? It has 2 ends. It has a back, it's like a backpack. It's carrying something, and it's got a reading section.
So the top end is the amino acid.
Good.
One is the anticodons.
Good. So we've got CAG as our first codon. What's the anticodon to that? GU. Very good, not T. And then C. GUC. So every tRNA molecule with GUC will have arginine. Does that make sense? That is how the ribosome has read the CAG and called upon arginine. It will recruit a tRNA molecule that will carry that arginine to the ribosome. Make sense? So right here, we've got arginine sitting here. Good. So now they've binded. tRNA's got an arginine. What happens next? The ribosome will then move to the... Yeah, so it'll move to the next position. But what you can see here, do you all see how there's 3 spots on the ribosome? Kind of like 3 cushy seats. And do you know what this is right here? That orange molecule. That is a tRNA. Do you see how there's another spot right here? Technically, the ribosome doesn't have to move straight away, but what it's going to do now is, since it's got 3 cushions for 3 tRNA molecules, without even moving, it can read the next codon sequence here. Okay? So it's going to read CUG now. That is a codon sequence. What is it going to do, Tashvia? It's going to call upon a tRNA molecule that has, very good, GUC? GAC, very good. Good. And for GAC, we'll always have lysine. Good. So now we have 2 tRNA molecules side by side on the ribosome. What's going to happen now? Pardon? Yes, the ribosome will form a bond between these 2 tRNA molecules right here, like this. That's a ribosome's job. What bond has it just formed? Good, a peptide bond. What bonds will break now? So I'll tell you guys that A, is it going to be B, is it going to be C? What bond gets broken now? Very good. Only A will get broken. So what's going to happen now is A will get broken, and that entire tRNA molecule will be removed. So what will end up happening now is we'll just be left with this. Okay? And what's going to happen now? Ribosome will read the next codon sequence. And let's say the next codon sequence is AAA. What tRNA molecule will be recruited? Okay. And let's say that codes for alanine. Okay? So this tRNA molecule will have alanine. Very good. What's going to happen now?
The lysine and the, what do you call that, are going to...
Alanine. Good, they form peptide bond. And what bond gets broken? Very good. Do you all now see the visual process that's happening? So then this tRNA molecule comes up, and look at what we're forming now. A growing polypeptide chain. That is exactly what the ribosome does. Okay? So you need to visually understand that. Now, you're not going to write this as your exam explanation. You're not going to draw 3 diagrams and label the bonds and talk about what's broken. So now my question is, now that you generally understand what's going on, how do you write this in an exam setting? I'll give you all a minute or so to think about this, because I'm going to ask a couple of you. I'll give you a very general question, and it will be describe 2 words, explain, translation. 3 marks. You'll never get a question this easy. So if you can ace these questions, then you have a really good foundation for doing the harder ones. So really think about what are the most important things I just learned here. Okay? One last question, everyone. When does ribosome know where to start? When does it know where to stop? I'll ask Guru.
There's the start codon and then stop codon. So the start codon, I'm pretty sure is AG.
Yeah, so there's variants, and typically you'll make the amino acid methionine, but you don't need to know any of that. So I think all you need to know for your exams is there is a set of 3 bases on the mRNA that is a start codon or tells the ribosome where to start. So the ribosome will first find that start codon, and then it will start attaching tRNAs, etc. Now, eventually, when the ribosome moves to the last 3 bases here, it'll reach what we call a stop codon. Okay? Does anyone know what that codes for? It's going to bring in a molecule that's not an amino acid. You know how one codon codes for one amino acid? In this case, this codon will not bring an amino acid. Does anyone know what it will bring?
Protein?
Yes, it is a protein. Very good. A molecule called the release factor. Maybe you can see that here. It should be in this diagram too. There you go. Look at that. Here's the final tRNA molecule with its long polypeptide chain, right? Just like how we spoke about. But the final sequence, this stop codon, is coded for release factor, which is a protein, completely correct. And what does release factor do? Yeah, it causes this polypeptide chain to break off the tRNA, and the tRNA be removed. And then it's done. That's it. Does that make sense? So now you know how the ribosome knows where to start and stop polypeptide synthesis too. Good. Do we have any questions before you answer my question? Yep.
Then we have the 5 prime.
Yes.
So because it doesn't code for that, what happens with the because there's no protein, like there's no amino acid being coded for, what happens after the whole process is
So the 5 prime, so during this entire process, where is this happening? In the cytoplasm, do you all agree? So that 5 prime cap and poly A tail are constantly being chewed on by enzymes in the cytoplasm, right? And the length of the 5 prime cap and the poly A tail will decide how long the mRNA sits in the ribosome. So think about it, if you have a lot of armor around that mRNA, it's going to exist and sit on the ribosome for longer, which means a ribosome can keep on making more protein. So that's how we control the amount of protein a gene makes. That's one of the ways we do that. Does that make sense to everyone? If it doesn't have much armor, aka it has a tiny 5 prime cap and poly A tail, you'll make a few protein molecules and the mRNA will be destroyed. If it has a huge case of armor at each end, you'll make huge amounts of protein from that one gene. Okay?
So if the mRNA is destroyed, does that cause a mutation?
No, it's just the protein stops being produced. Yeah, and then RNA, then transcription processes start all over again. Yeah. Good questions. Lookman, what's your question?
Why do they use an adenine tail? Like, why do they use adenine as a base instead of all the others?
The tail is so it's a 5 prime poly A tail is adenine. You are right. That tends to be one of the more stable bases. That's a bit more resistant to degradation. That's why it's also in the mRNA strand in general. I guess to answer your question, we don't really understand. This is more from microscopic electron microscopy studies where we find out what the tail was made out of. But it's kind of like a redundant set of bases. So you could have picked anything. It could have been a T, a C. I think it's just evolutionary chance that we use adenine as our tail.
Yep, thank you.
Good. And rest of the online students, when I ask you a question, make sure you answer on time, please. So Kushi and Raya and Harsha. Good. Okay, so 1 minute before you answer my question, which is up here. Feel free to discuss among your peers, but I want a good answer. Raya, can you hear me? Kushi, is your audio working?
Yeah.
Good, good. Harsha, is your audio working? Harsha and Raya, can you please make sure your audio is fixed ASAP? I will be asking more questions.
Excuse me.
Yes?
Do you know how you provided exam descriptions?
Yes.
Do you think do you think we should know those descriptions for the exam, or
As in, should you know those descriptions?
yeah, like should I use that description when I'm writing an exam?
Use a template. So never memorize, understand
Yeah.
the point of that exam answer, and that's exactly what I'm talking
Yeah. Okay. Yeah, that's what I meant. Yep.
Okay, let's start. So, I want you to close your books. You're not going to read off anything. The thing you realize is if you can say it, you can write it so easily, right? Because you think fast, you write really slow. Okay, so we'll start with Alicia. What's your answer? Loud and clear, everyone.
The ribosome moves along the strand and it eats the lungs, and then complementary tRNA is the complementary tRNA binds to the codon and anticodon. It forms and up creating an amino acid. And then the multiple amino acids form a polypeptide bond, and then it's eventually.
Good, I'll stop you there. We'll go to another table. What do you all think of that answer? Is there anything that could be added?
Maybe just you had a quick one sentence explanation of what translation is as well.
So, actually, we'll start with you. Give it a go. What is that one line?
So I said that Translation is the process that forms the bridge between mRNA strand and the creation of amino acids that then form a polypeptide chain.
Good. One thing is, for science, I want you all to be less Englishy in the sense that, you know how you mentioned a bridge?
Yeah.
It's a bit metaphorical. You need to be as direct as possible, right? The mRNA sequence enables the formation of an amino acid. Ria, what else would you want to add?
I was just saying the antigen, maybe just briefly mention that in tRNA they have the antigens and the amino acid is already present, and that's when it binds the amino acids already present.
Yeah, good. She did mention amino acids eventually, and I thought you were going to forget anticodon, but you came back to that, which is very good. I think that's a good answer. There's only one thing that I would like you all to say at the very end. Did you all hear thus as a result of due to in her answer? What is an explained question? What does that entail? Cause and effect, right? So if you aren't using those terms, you haven't explained it, you've described it. This is to do with exam technique, which you also need to think about, right? So when it comes to a question like explain translation, I'm going to ask one more person. I want to hear thus, due to, as a result of. Okay? Causation. Good. So I might ask one more person. Pranav?
Translation is the process of translation is a process where amino acids are formed. So this process is started with the mRNA strand, which enters the cytoplasm and attaches to the ribosome, which amino acids are created through the help of tRNA, providing anticodons to the mRNA strand, thus providing a strand of amino acids which form peptide bonds.
Good. I paused you there. Do you see how it's challenging? I think for the people I've asked, you feel it's pretty hard to go off the top of your head, right? That's how you should be studying. Okay? That is the best way to study biology. If you just write, you highlight notes, the worst thing you can do. When you do things like this, you understand what you don't understand. And that clarifies you on where you should be studying, right? So very good. But things I don't want you to use again is like with the help of. A bit of Englishy terms. I want it to be very direct. Okay, what exactly happens? Well, the anticodon of a tRNA molecule binds to each mRNA codon sequence. This enables adjacent amino acid ends to bind via peptide bonds, forming a polypeptide chain. Something like that. Okay? Yes?
What would we say is the actual definition of translation?
Okay. Good question.
In the first sentence, so you have the clear definition, and then you explain the
Sure, I would say you don't even need a definition. If it's a 3-mark question, you don't even need the overall point, but it's good to have. I would say if it's 3 marks, 3 points. If you're writing more than 3 sentences, you're writing too much, you're wasting time. Same thing, the number of marks tells you how many lines of points you should write. Okay? So if I were to give you a definition, it would be translation is a process by which an mRNA strand is converted into an amino acid sequence through the use of ribosome. That's quite literally what it is. Or you could change amino acid sequence to polypeptide chain. That would be more correct. Okay? Good. Now, what you saw me writing here was buzzwords. As a marker, that's what I want to see, right? I want to see the words codon, complementary, tRNA, and anticodon. And before you're all confused, because I still haven't given you an exam answer, this is my exam answer. So I wrote that for you. Okay? Very excited, Georgina. This is my exam answer for translation. Now, I would not ask you to memorize it. This is not for you to memorize and regurgitate. It is for you to understand the key points. Now, I think I stressed this. It's a ribosome that moves along the mRNA. That was important, and that's how I start off. Okay? I've used the word complementary. So what I'm doing now is I'm going to highlight the key words you must have. Good. You see how direct this is? I wasn't vague, it wasn't metaphorical in any way. I literally am explaining exactly what happens. Complementary tRNA anticodons are added to each sequential mRNA codon. That's it. Okay? Now, the ending part is quite unique. What I'm trying to do in my explanation is this. Now, if I told you A equals to B, B equals to C, what's the logical conclusion we can make? If you do general maths, you'd get that, right? Very easy. Now, A is your mRNA sequence, right? It's your mRNA codon sequence. What does that code for? Or what does that recruit? It recruits a tRNA molecule which has a complementary anticodon sequence. Now, what did I tell you? For every single anticodon sequence, there is a specific amino acid on the backside. So for every single anticodon sequence, there is a specific amino acid. So what's the end result? Yeah, thus the mRNA codons are determining the amino acid sequence. That is what I'm trying to explain in my explanation answer. And that is what this entire section is all about. Have a read of it. I've said as tRNA is specific to the mRNA sequence, and since tRNA is specific for an amino acid sequence, the mRNA codon sequence will determine the amino acid sequence of the polypeptide chain. Okay, so that is my final point that I'm trying to get across. So if you take anything away from your translation answer, it should be this. Does that make sense? Okay, now we've gone into a lot of detail on transcription translation. We're going to move on now. But do we have any questions at all? Questions? We'll do some exam questions later. But for now, I want to move on to more content. Who's learned this at school? Which hand up? Yeah, made sense at school? No, not really. Went a bit fast. Do you understand it a bit better now? Makes sense? Any other questions at all? Ask anything on your mind.
Yeah, sir.
Yep.
Could you please explain that, what you circled again? I don't really...
This part. Yeah, sure.
That.
So what I'm trying to explain, Lookman, is the mRNA sequence, each codon, right? Remember, going back to our diagram, have a look at this. You see how every single, I'll do this in this color, every single codon sequence, which is what I've shaded in blue right here, is determining the complementary anticodon sequence. Do you understand that?
Yep.
And every single anticodon sequence is specific for one particular amino acid. So I'm trying to explain that since this mRNA sequence is determining the tRNA sequence, and since the tRNA sequence is coding for the amino acid sequence, we can thus say that the mRNA sequence is directly determining the amino acid sequence.
Oh, right. Okay. Now I understand. Yep.
Good. Does that make sense to everyone? Yes. It's quite warm in here. Yes, that's the questions I want you all to do, which is in your booklet, don't worry. But you're a very good plan. When I was brought up, this is the way they're going to test you. So we'll do that now, in fact. Okay? It's a very good question on this. Hopefully I've got it up here. I'll scroll down and see if we've got a question on that. I'll bring it up for all of you shortly. Okay, so maybe after the hour break, I will bring up that question. It's in the HSC additional questions booklet. Have you all seen that booklet? No? So there's two ways you can access it. So if you go on the edX website and you go to HSC Biology, you'll see HSC additional questions booklet. That's a NESA 80-page document that you should be doing right now. It's probably the best time to be doing it, right? Because it's HSC level questions for every single topic. For every single topic, for every module, they've got 2 or 3 questions for you to test your knowledge out. These are all exam-oriented questions, so they'll be super helpful for you, okay? So maybe write that down to start doing these questions as you learn through the content, okay? Good.
Excuse me, sir.
Yep.
So, when doing questions, how do we, say if we get a question wrong, how do we utilize the sample answers? Like, how should we like just add it to our notes? Or
Who's seen the sample answers? Who's done questions and seen sample answers? Yeah. Now, I'll tell you all this. Who's also writing module notes? You don't have to. I definitely did because I didn't have notes like this when I was in Year 12. You guys have pretty much everything in front of you. So if you think that you back yourself to do more questions and save time instead of writing notes, go for it by all means. Okay, but it's all up to you. Is anyone handwriting notes? I wouldn't do that right now. And the reason is you all have to realize you don't know anything right now. You don't know how to write an exam answer. You barely know the theory. So whatever you write in your notes is not going to be top notch. You have to constantly revise it. So if you are planning on writing your notes, which is what I like as well, I prefer that, I would handwrite or type summaries. I would do questions. I would copy paste sample answers back into my notes. And then when I finish the module, I get like 5 or 6 palm cards and write the key most important things. Does that make sense? So that's how I would take about writing notes if you want to handwrite it. Okay? So to answer your question, Lookman, I copy pasted sample answers into my notes. Right? They're very good. The sample answers can sometimes be very brief, but they always will have everything you need to answer that question. So you look at the wordings, look at the sentence structure, copy paste that into your answers. Yes? Yeah, so again, on the edX website for the trial papers for 5, you don't need to be doing them right now, but they all have sample answers that you can just copy paste and put into your notes. And it's the same for the HC additional questions. They all have sample answers, the band level of that question, which is really good as well. Yeah. Yes. So this is the other thing, right? One of the reasons why I did well in biology was not just because I did tons of papers. You realize all of you will do tons of papers. You all max out all your papers, all your peers will. You need to be very, very meticulous in marking your work, right? So rather than giving all your work to your teacher to mark, you should master becoming a marker yourself. Because think about this. In the HC exam, you'll finish your exam about half an hour early. In that half an hour, what I did was I opened my paper from the beginning and I looked at it like a marker. I scrutinized every single line and I picked up about 10 marks. If I hadn't done that well, I would have got nowhere near the mark that I ended up getting, right? So you need to really perfect how to be a marker, pick up the key points and questions as well. So I would say, look at marking guidelines. You can add them to your notes as well. If there's a really good question, as a really good structured marking guideline, copy paste that into your notes. It tells you what the most important things you need in your answer are, right? So I'd say that's equally important. The syllabus is very good for structure. It's because the syllabus, every question in your HC exam comes from the syllabus stop point. So if you can close your eyes, have a mind map of the syllabus, whatever question comes in front of you, the first thing you can do is think, well, what syllabus stop point is it coming from? And then you know exactly what you need to write to get the marks. Because sometimes in bio, what happens is when you get a vague question, the student doesn't know what module or dot point it's from. They write whatever they think. Sometimes it doesn't align with that dot point and they lose marks. So that's beneficial there. Good. All right, let's move on. Let's now go into dot point 3. Assessing the function and the importance of polypeptide synthesis. Now, whenever you find out how important something is, take it away and see how that changes all the other variables. So if we didn't have polypeptide synthesis, what would be the implications? Let's say, I was an X-Men mutant and I could stop all your polypeptide synthesis. What would happen to you? Yeah. Any ideas? I want to just think critically in terms of what proteins you know. You know proteins are important in muscle tissue, so you'd stop making muscle. But that's not going to immediately cause you to waste away and die. What's going to happen to you?
I did this for sickle cell.
Yeah. Yeah, no more hemoglobin, so you get oxygen in, but the oxygen can't move around. Hemoglobin is kind of like a Uber vehicle that carries oxygen around to the body and delivers it, right? Oxygen is like the Uber Eats, it's the food, the nutrient. Your hemoglobin carries 4 oxygen molecules and will transport it around the body in the circulatory system. So you don't make hemoglobin, you will lose oxygen carrying capacity, your cells will start to die because they don't have oxygen, which is important for what process? Bumbling. So we have a clear answer? Why do you need oxygen? Why are you subconsciously just moving a diaphragm muscle, breathing in and out? Yes, cellular respiration is the answer. You needed to make ATP. ATP is a currency that makes the internal body function. Good, you were about to say something? Yeah, is there any more immediate effects if I stop your polypeptide synthesis? This is teaching you what polypeptides are used for. So we just went through muscle tissue. We went through functional proteins like hemoglobin. I'll write this all down, but what else? Good, right? So all your enzymes you'd not make anymore, right? And so you wouldn't be able to carry out chemical reactions at body temperature. 37 degrees is lousy. It's a very low temperature. Reactions happen too slow for life, right? Nutrients don't go in fast enough, waste builds up, you'll die. So the way we bypass that and engage in fast reactions at low temperatures is through enzymes. Okay, good. Who does chemistry here? Yeah? Who's seen something blow up before? Surely all of you, right? Those are combustion reactions. They release a lot of energy. When you engage in respiration, that's a combustion reaction. It uses a ton of energy, it releases a ton. The way we do it at very low temperatures is through enzymes. Okay? Good. What else are proteins used for? Yeah, very good. So all your hormones would go, you stop making estrogen, you'd stop making testosterone, right? But that results in a lot of effects. You would lose your male or female characteristics. What else? So I think I'll start writing a few of these very important points. But we need to categorize this. You see how we're kind of sprawling everywhere? In your heads, what were the two categories I told you need to have? Good. Functional and structural. So I'm going to start listing them because your notes have everything you need. But we'll start with functional. I want to hear enzymes. And for structural, I think muscle tissue is important. Do we know what two proteins make up muscle tissue? Actin and myosin. And pretty much it's like a pushing, pulling motion, right? With the action of ATP energy, the actin can pull on the myosin or contract a muscle. And what does that do? Think about your finger. Bend your finger for me and you'll see actin and myosin in these lumbrical muscles are contracting, and that's enabling that finger to contract and move inwards, right? And how are you controlling that? Your brain is sending nervous signals to those muscles to do that action. It's pretty cool. Good. Who's touched something really hot before in their life? What do you reflexively do? It wasn't even your brain that made that decision. It was the spinal cord. Because if you had to go, the signal had to go up to the brain that you felt pain and then go back down, you'd already burnt your hand, right? So it's a spinal cord reflex that sends signals to the muscle to contract the actin and myosin and pull your hand away, right? It's quite interesting that muscle tissue is so important for survival. Good. What else? We mentioned a few other things. So enzymes, I think hemoglobin should kind of be its own category. It's so important. Hemoglobin is a very important protein. Oxygen carrying protein. What else? When you're all thinking, what are you doing? You're releasing essentially what you are, everything, your identity, your thoughts, your everything about you can be broken down into a set of nerve cells that are just firing or not firing. That makes up all of your thoughts. Right? We're very complex people, right? We have very nuanced thoughts. In the end, it's just firing and non-firing of nerve cells, right? Do we know what nerve cells release to talk to one another? Neurotransmitters. And guess what? Neurotransmitters are proteins. There you go. So the first thing that would happen if I stopped all your protein synthesis is you wouldn't be able to think. You wouldn't be able to release any neurotransmitters. And that would probably be one of the first implications. Good. And maybe I'll give you a name. The most common neurotransmitter, there's so many, but the most common one that all of you are releasing right now when you're thinking and focusing is glutamate, the protein. Good. What else? I want you all to think critically. What other proteins do we have? What are some of the structural proteins? Women's skincare products. What do they have? There you go, right? You already knew this. You're teaching me. Collagen. Collagen is a protein that is found in skin, in connective tissue. It's found in your bones. It's found in your cartilage. And it's very important for the non-cellular component of bone, cartilage, and tissue. Okay? Very good. Good job. Collagen. Okay.
Excuse me, sir.
Yeah.
Would keratin be one?
Very good. Keratin is another structural protein. It's in your hair, your nails. Keratin is an armor. It literally is, right? Your skin, the reason it's different to the inside of your mouth, which is, moist, but it's also prone to damage, is because skin is reinforcing an armor of keratin. That's why it's very rare to get very sick from skin infections, but much more common to get sick from intestinal or internal infections, right? Very good. Is skin alive or dead? Yeah, the outer layer you're touching is all dead. The inner layer is alive. You can't see that. Has anyone scratched their skin and they see white sometimes? That's, you've gone through the entire skin layer, you've gone to the fat underneath. So the layer just above that is the live skin cells. Good. What else? A bit more.
Calcium.
Calcium is an ion. It's not necessarily, but calcium is used to enable muscle contractions in the cell.
So
Yeah.
for a functional ferritin, would that be a
Ferritin?
protein?
Good. Good, I would say it definitely is, but I wouldn't go into that much detail. So for everyone who heard this name of the protein, ferritin was something that was just mentioned. Ferritin is kind of like a sack of iron in the body, right? So any extra iron in the body is stored in little sacks. You know the sack that Santa Claus carries on his back? That's how you think about it, right? That's ferritin. It's a carrier of iron, so that's what it does. So if you're anemic, you have a lot of ferritin, and we can check for that at hospital. Good. I'd say that's quite expansive, but let's go to our answers here. So you can see how I've broken it down, and this is going to direct your exam answer. When you talk about proteins, structural, functional, and then you can go through an example. Now, I've got one example that I've gone through in detail, which is here, thalassemia. I guess I'll go through that with you right now. Now, a bit more detail on hemoglobin itself. Hemoglobin is made up of 4 polypeptide chains. So that's 1 polypeptide chain, 2, 3, and 4. Okay? I'm going to come back to this because this is very important. At the center of each polypeptide chain, we have a metal. Do we know what metal that is? Very good, right? That makes sense because if you're iron deficient, you get anemia, which is a condition where you can't carry as much oxygen, right? Anemia is very interesting. Is anemia a diagnosis or is it a symptom? Actually, a symptom. Anemia always has an underlying cause. Is it blood loss? Is it bone marrow problems? Is it iron deficiency? Is it cancer? Cancer can sometimes just present with anemia, right? So anemia is a symptom that should be explored further. We'll talk about anemia later, but good. And every single ion can carry 1 oxygen molecule. So how many oxygen molecules can 1 hemoglobin carry? 4. Very good. Now, hemoglobin, think of it like one protein like this. Now, have you all poked beanbags and felt what's inside a beanbag? That's pretty much what a red blood cell is. It's a beanbag full of hemoglobin, right? So the red blood cell is the actual sac. Inside of it is full hemoglobin. Each molecule is carrying 4 oxygens. That's it. Does it have a nucleus, red blood cells? Why not? Very good. It's all about space. Carry more oxygen, which is better. So they actually get rid of their nucleus as they mature. Very good. Good job. Now, thalassemia is a condition. Does anyone have thalassemia here? Yes, you do? We've got a survivor. It's not a fatal condition. A lot of people have it. I think a huge portion of the East, South Asian, and Middle Eastern population have thalassemia. And you can have it to differing degrees. But essentially, what thalassemia do you have, Georgina? Alpha, are you alright? Okay. Do you have alpha or beta thalassemia? Not too sure? Go home and find out. But if I had to bet, I'd say you had beta. So minor is to do with the severity of the thalassemia, not the type of thalassemia. So everyone, we call these chains alpha chains and beta chains. So we have 2 alpha chains and 2 beta polypeptide chains. Now in thalassemia, there is a mutation in the gene that makes one of these chains. That's it, right? So what happens in beta thalassemia, can anyone guess? What's the issue? There is a mutation where you don't make one of the beta polypeptide chains, right? And since you don't make it, do you have as much functional hemoglobin? No, you don't have functional hemoglobin. So you can't carry as much oxygen. If you can't carry as much oxygen, the oxygen doesn't go to your cells to make ATP energy. So what symptom would you have if you don't have energy? Fatigue, which is essentially anemia, right? The way we diagnose anemia is we look at the hemoglobin. We measure the patient's hemoglobin in their blood. If it's low, we say anemia. Does that make sense? So if I had to define anemia for you, beta thalassemia is a gene mutation which results in decreased beta polypeptide. So what would alpha thalassemia be? Same thing, right? Very good. Decreased functional hemoglobin. Good. So now we've just linked the gene mutation to the phenotype, which is a physical condition the patient will have, right? Physical condition is they have fatigue. They can have breathlessness. Anemia, does anyone know any other anemia symptoms? I'm sure some of you would have anemia here. It's a very common symptom, especially if you have a vegetarian diet, for example, much more likely, because red meat is rich in hemoglobin. Yeah, but is that a symptom? A symptom is something that you feel. Can you feel yourself being iron deficient? Not really. Feel the fatigue. Yeah, you get dizzy, right? Because there's not as much ATP, there's not as much oxygen going to the brain. And less oxygen to the brain make you feel faint. Very good. Anything else? You can. One weird symptom that I know people have is the tendency to eat ice. Yeah, so a lot of people with anemia, they have this weird tendency to want to eat ice, which is, I can't explain that, but it's a very interesting symptom. Yeah, that's another symptom of anemia. Anyway, thalassemia is a cause of anemia. Does that make sense? Anemia is a symptom. Very good. Let's move on to the next dot point. This is a very important dot point. Could I get... maybe in 5 minutes I'll give you all a break as well, but could I get... Nisha, can you read out the dot point for us? Sub-dot point 4.
Assessing how genes and environment affect phenotypic.
What is this question actually asking? Like, it's a very interesting question. Someone asked me the other day, how much of depression, how much of schizophrenia is genetic? How much of it is environmental? It's a cool thing to understand, right? How much of, is there any other diseases which we're interested in knowing how much of it is genetic? Heart disease, right? How much of it is genetic? How much is in your control? And how much are you just born with that or good luck? Okay? Yeah, diabetes is another big one. Now, does anyone know? Diabetes is one of the leading causes of death in the entire world, right? Because it causes heart disease. It causes, vision problems. It causes stroke. Is it genetic or is it environmental? You have to pick. So I'm asking you a percentage. Pardon? Yeah, I'm asking for a number. What percentage of, I know there's type 1 and 2, don't complicate it. They're similar, actually. Which one? Anyone think it's more genetic or more environmental? It would sound like it, right? You eat sugary food, that can give you diabetes. That's probably what you all learned. It's actually very much genetic. And guess what? The ethnicity groups in this room are all high risk for diabetes. Yeah? It's interesting facts, which we'll talk about later, but South Asian and East Asian individuals have smaller pancreases. And pancreas is the organ that releases insulin, which controls your blood sugar. That's because, and evolutionarily, we didn't eat as much carbohydrates as Caucasian people. So our pancreas has evolved to be smaller. So then you migrate to, Europe or Australia where there's, sugar, sweet, high-calorie burgers everywhere. The organ can't handle that much sugar, and it can't control it that well. More likely to get diabetes. Yes?
Are these changes due to dyslexia genetic?
Yeah, see, that's another very interesting question. So to answer your questions, diabetes is about 70% genetic, which is a bit worrying. So if you have a family history, ask mom and dad, grandparents, you're at risk, right? Same with heart disease. Yes? Schizophrenia. Okay, so for everyone to understand schizophrenia is where you have delusions, hallucinations, so it's usually auditory hallucinations, and you have disorganized thinking. You can't think straight as well, right? And it's a really debilitating illness, and it's 50% genetic, right? Now, I have a question for you, because this is what this exam dot point is about. How do we get these percentages? It's obviously studies, but I'm asking you, what's the design of the study to get you that percentage? Well, yeah, obviously your data collectors, you're getting a number, but I'm asking you, what would the study look like? Yeah, close. Close. Yes, you guys are all very vague. I want you to think what specifics. If I gave you $1 billion to create a study to look at what percentage of ADHD is genetic, what would you do? Yeah, but then pedigrees, you can't get numbers directly. Anyone know? To find out how much of a disease is genetic, it's actually very simple. You get twins. Same genes. You look at what percentage of twin 2 has a condition that twin 1 has, given that they're monozygotic or identical twin. That's it, right? So you in your exams will definitely see a bunch of questions called concordance studies, right? Where they'll show you a table of twin 1 and twin 2 and their percentages. And they'll ask you, which of the diseases is most likely environmental or genetic? And you will have to come to the logical conclusion. We'll do those questions soon. But that's very important. What I'm just going to write here for you is concordance studies. Have a read of concordance studies in your own time, and I'll give you a few questions to do on them as well. They're very high yield. If they are to test this dot point, 50% likelihood they will give you a concordance study graph or table to analyze. Good. All right. Let's move on. We're going to go through some diseases that are very much environmental. Okay? But the end answer for this dot point, all of you, what you need to say is that all diseases have some genetic basis and environmental basis, and it varies. And in your exam answer, you want to give conditions that have both. Okay, so I've selectively picked a lot of high yield conditions that have both, that have come up in tons of exam papers as well. The first one is this condition called PKU. Okay? Fancy name, phenylketonuria. Let's break it down together. Phenylketones are a type of molecule. What's urea? Urea means urinating, so it's found in the urine of these patients. Okay? But that's what the disease means, just so you understand it generally. Now, to know what it's about, in phenylketonuria, it's a genetic disease where you have a knocked out gene that makes this enzyme, phenylalanine hydroxylase. Okay? For those of you who do chemistry, hydroxylase, hydroxyl groups, OH groups, it just adds OH groups to molecules. That's what a hydroxylase does. Now, in this disease, a gene mutation knocks off phenylalanine hydroxylase. This was an exam question. What is going to happen the levels of this chemical and the levels of this chemical in a normal versus a diseased individual? This is the 2020 exam choice trial paper. Phenylalanine? Very good, right? And you'd say a normal subject relatively would have more tyrosine, less phenylalanine. Very good. So, do you all agree, the issue here is we're having too much of this chemical now, because it can't be converted into tyrosine. Now, too much of any chemical in the body is bad. So what this chemical in particular does is it goes to nerve cells and kills them. So what symptoms do you think a patient would have with this disease? Yeah, they could have, sensory impairment, could be. What else? Think about the brain. If you kill brain cells, yeah. Intellectual disability, that's what will happen, right? What else can happen? Seizures, right? When you have damage to brain cells, they start firing in a very disorganized fashion. They can give you seizures. So we typically see this in Caucasian babies, blonde hair, blue eyes, fair-skinned babies. They can get this gene mutation. So build-up of phenylalanine causes damage to nerve cells. Damage, nerve cells. Now, you're not doing medicine, so you don't need to know all the symptoms. I just remember one, intellectual disability. Okay? Good. So my question to you is, how do we prevent a poor kid from getting intellectual disability? Imagine, you're a doctor, baby's born, it can get intellectual disability from this condition. How can we prevent this using your understanding of HSC biology? You go think back to what you learned in HSC. What can you do for this baby? You all have the answer. I've kind of been feeding to you this lesson. Phenylalanine is an amino acid. Where do amino acids come from? Lookman, where do amino acids come from?
The food we eat.
Yes. So what can we do for this child to prevent it? There you go. You're already ready to be a doctor now. Very good, right? So all we need to do for this child is restrict phenylalanine from their diet, and they'll be completely normal. Who drinks Coke? Check the Coke can. You'll see in fine script it says contains phenylalanine. The reason they do it is for these mutations, right? The people with these mutations can't have Coke. That's not the only thing, though. It's not just Coke is depriving the diet. There's many other foods. There's a lot of different foods. I don't know the entire list off my head, but there's certain meats that you can't have, which are rich in phenylalanine, certain dairy products. So they have to have a very specific diet. They know what foods they can and can't eat. But that's all you need to do. So my question to you is, do you all agree this disease was genetic? But do you all agree that phenotype, which is a physical characteristics, intellectual disability is part of phenotype, was ultimately decided by the environment? So this is a really good disease to highlight that genes and the environment affect phenotype. Good. Any questions? Very good. Good job. On that note, before we go through, in fact, I'm not even going to go through the second one, which is hydrangeas. You can read that in your own time. This is basically flowers that grow different colors in different pH, right? So they grow blue in acidic soil, they'll grow pink in basic soil. You can just read this in your own time. It's not too interesting. Have one really good example, and that would be good enough. All right. Have a break, everyone. Stand up, stretch. We'll recap at 12:30. Feel free to go get some water if you'd like, use the restroom. Okay, now, before I go back to the immediate concepts at hand, any ideas about this question here? Now, the thing is, your brain and my brain can't wrap 7.8 billion, right? Sydney looks like it's pretty overpopulated. If you go to the CBD, it's pretty packed. And Sydney is only about 26 million people. That's 7.8 billion on Earth. So any ideas how we can solve this issue? Because right now we're using 4 planets worth of resources, and that is why Elon Musk, for example, is so keen on going to Mars. Not just to have a party there, but to get resources and ensure continuity of our species. So, any ideas? I think the thing you'll realize is after you finish high school, you don't get asked closed-ended questions like explain transcription. You get very big questions like this that you need to be answering, even in your work or whatever you do. So thinking critically is such an important skill for life. So anyone have any ideas on what we could do? We'll go table by table, yes?
Increase access to content.
Very good, amazing. I think that you straight away hit the nail where it happened. the question you should have asked yourself is, well, where is the 7.8 billion? Are they all in developed countries or developing countries? And you realize the majority of the world population, I think only 1.7 billion is in developed countries. The majority is in developing countries. And poor health literacy, no access to contraception means they have many children, right? As a result, it increases the global population. So then we need to think about how do we fix the issue there first, right? So the solution, number one, is access to contraception in developing countries. What else could we do? Pretty cool. Good. Hey, look, it's put on the table, but we're not gonna do that. We're not gonna do policy rules. Anything else? No, yeah. So see, that was the logic behind the whole Avengers and Thanos and the finger snap. Like it makes sense in the sense that you need to reach some kind of equilibrium, right? In fact, one of the best things that happened to the planet, in terms of nature, we're not talking in terms of humanity, was the bubonic plague, which happened a few centuries ago. And it resulted in a huge portion of humanity being wiped out. But in terms of the planet's health, significantly improved, which is very worrying, right? That diseases are happening every time the population reaches some kind of peak. So what else could we do? Yeah, right. So maybe diet. Switching to a more plant-based diet is one of the reasons. I don't do that, but just a point that I'm mentioning. It definitely does make sense. Anything else? But I don't think plant-based would fix the population issue, right? It would fix the resource use issue while having the same population. So now do you see how we're going into mutually exclusive categories? We can keep the population the same, minimize the resources used per population, or we can drop the population number. You see how there's categories to thinking there? Very good. I'm looking for one more thing. Yeah. Yeah. Yeah, you would say that. You definitely could argue that the planet and diseases will find a way to reach an equilibrium. As in nature, right? Every single population other than humanity is controlled by evolutionary forces to maintain a population size. So you could argue that, but we might get extinction of humanity if we wait for more pandemics to hit us, right? Anything else? Yeah. Okay, so being more stringent in terms of legal parameters, sure. Anything else? Well, one very interesting thing is when you educate and empower women, right? What happens? Focus on career, equal opportunities. And what do you notice in metro countries, right? Women have children later and they have fewer children because they have so many other things to do. And so that's one of the major solutions to maintaining the population size as well. So that's something that Bill Gates and other philanthropist billionaires are trying to do, which is to educate and empower women, which indirectly reduces the population size. Pretty cool ripple effect, right? Good. I want you all to be looking into things like this, not just always studying, because then you don't have any character or any thought outside of HSC. You don't want that to become your identity this year, right? So make sure you guys do other stuff outside of that, and also read widely and do things you like. Get good at it. Okay. Good. All right, let's move on. Now, this is your next dot point. The different structure and functions of proteins in living things. Now, we've gone through the function of proteins, right? We said it can be structural, functional. We went through the different categories. What we haven't gone through is the different levels of protein structure. And this ranges from primary to secondary to tertiary to quaternary. And they love asking these questions through diagrams. Now let's go through them. What do we all see here? What are you looking at? These are amino acids, so what would this chain be called? Very good, you all understood that. Now a polypeptide is the simplest level of protein structure. Do you all agree? Good. So we call that a primary protein. All polypeptides are primary proteins, right? They're a chain of amino acids. That is all a primary protein is. The next to primary, it's already been written for you, but you can highlight, it's just an amino acid sequence. That is all a primary protein is. Does that make sense to everyone? Good. Now, have a look at this. As we go to secondary protein, what is happening now? We're still looking at one polypeptide chain, everyone. But what's happened to that polypeptide chain? It's formed shapes. Do you all see that? One shape is a helical arrangement, similar to DNA. Do you all see that? Helical. And one looks like a sheet, kind of like in old houses, the roof that you'd have in old houses, right? So when we talk about secondary structure, there are only two types, and that's why you need to know it so well. There's what we call an alpha helix or a beta sheet. If you don't mention these two words, you will get no marks for secondary structure, right? So secondary structure is when we get a bit of folding of that amino acid sequence to form a more complex shape. Now, why does it fold? To answer that question, what is the bond between bases in DNA? Very good. Hydrogen intermolecular bonding, right? So hydrogen bonding is what causes the 3D shape of the amino acid sequence. Have a look here. What will happen is this amino acid sequence will form a hydrogen bond with this one. This one will form a hydrogen bond with this one. And do you see how that's going to force it into a unique shape? Now, ultimately, they will always form one of the two, a beta sheet or an alpha helix. And the reason that happens is due to what bonding? Hydrogen bonding. Easy. Any further questions? No? Makes sense? Remember, we're still talking about one polypeptide chain. So important. Yes. So all proteins will have one or the other, and it will be the normal form. So sometimes a protein goes into a beta sheet instead of an alpha helix. That'll form a disease because it was meant to go into the other shape. Yeah, so they don't randomly go into either one, they'll always go to one or the other, depending on what protein we're talking about. Okay, good. Good question. Now, what do we see here? Further folding, but do you see it's more complex three-dimensional folding now? So here we had almost linear folding, where it formed a helix that went in a straight direction, where it formed a sheet that also stretched up in one direction. Now, if we get the sheet or the helix, and then we fold it into a three-dimensional object, like a beanbag, right? Something that's folded multiple times in on itself to a 3D shape. That is what we call tertiary proteins. The tertiary proteins is further 3D folding. And this happens due to stronger bonds. So not just your hydrogen bonds, which are really weak. What temperature do we need to break those hydrogen bonds, by the way? Not 37. Told you all a number, and I told you I would ask you lessons later.
90 degrees.
Very close to 95 degrees, but I'd give you that. Very good, listener. Good job. So 95 degrees is the temperature in which you can break those hydrogen. If you don't have 95 degrees, what can you use? Cold? What enzyme do you use to break DNA? Helicase, very good, right? So this is how we're linking different parts of the logic together. Good. So tertiary proteins have much stronger bonds. You just need to know one example. There's many types of bonds you can have. These are what we call covalent bonds, right? An example that we can have is what we call a disulfide bridge. Again, it's never going to be part of a marking criteria that you mentioned this word. So I wouldn't commit it to memory. If you do, remember it in an exam setting, good. Okay? So disulfide bridge is just when you form a bond like this, and there's a sulfur atom that helps that bond strengthen itself. Don't commit it to memory. Understand. Okay? Any questions so far? Good. Now, this is arguably the most important part of this entire dot point. The defining feature of a quaternary protein is more than one polypeptide chain. Did you see primary, secondary, tertiary? How many polypeptide chains are we dealing with? One. The second you add more than one, you are immediately what? A quaternary protein. Very good. So, coming back to my question, what would hemoglobin be by that logic? Ordinary. In exams, they've literally given you an image of hemoglobin, and you had to identify what level of protein structure that was. And a lot of students didn't get that correct. But if you remember this one point, very easy. Okay? Any questions so far? Good. Online students, Kirthana, Kushi, Lukman, any questions at all before I move on? Yep? Yep. Happen? Yeah, it's catalyzed by enzymes. There are other enzymes that come along and do that. Sometimes it will be just due to the fact that there's unique amino acids that can form those bonds because of the side atoms that they have. So that's very complex. It happens because there's so many enzymes in a cell that do their own processes, bind to things, cause actions. What we're doing in HSC Biology, we're going very high-level overview on these things, right? So if we actually zoomed into the detail, you'd see there's so many enzymes involved even in modifying protein structure. Okay, we're kind of skipping all that, but that would be the answer. Good. All right. Again, here is a bit more detail on functional and structural proteins. I guess I'll just name drop a few things. Who did the catalase enzyme practical in year 11? A few of you, surely, right? I did it. I remember doing that like 7 years ago. Why did you learn about catalase? What does catalase do in your body? Speeds up things, it breaks food. Is that it? What if I told you it's literally your savior from cancer right now? So all your cellular processes, they create hydrogen peroxide. Hydrogen peroxide loves ripping electrons off things, causing mutation, and that leads to cancer. Catalase is present in your cells to immediately destroy that hydrogen peroxide, right? So who's heard of antioxidants before, right? It's marketed a lot in fruits and skincare products. Catalase is an antioxidant. When you rip electrons off things, you are called an oxidant in chemistry. So an antioxidant prevents those things from acting. Okay, so that's a bit of chemistry explaining a layperson term that you would have heard many times. So catalase is very important for your survival. So that's another functional enzyme. Okay. Actin-myosin, I guess I'm zooming into things here, but look at that muscle, and when you zoom into the muscle, what essentially is happening is, hopefully you can see the word, this is myosin, and this is actin. And do you see how the myosin has heads? That's the myosin. It has heads that can hold on to actin and pull, right? So under the action of ATP, when you give ATP to the myosin, it'll pull the actin towards it. And what would you see in this person's arm? Contraction of the arm. Makes sense, right? So that's how muscle contraction works. Very good. All right. Happy to move on? And that takes us to our next inquiry question. Good. I'll let you all read this one. So, Kashfia, can you read out the inquiry question for us? Open question to all of you. How can it be compared? Yeah, right. So you could read DNA to compare similarities and differences. Very good, which is what we're going to be looking at very soon. Anything else? Yeah, you could look at ancestry. Very good. Anything else? Yeah, very good. So you learn about pedigrees and another thing called Punnett squares, and you'll learn when to use what. A lot of students are confused about when you need to put a pedigree, when you need to do a Punnett square. They're very different things. We'll talk about that soon. Good. Let's start with this dot point here. Now, this we're obviously not going to do a practical investigation, right? But who's done a modeling experiment or done it's practical at school where you might have been assessed on meiosis and etc. Who actually modeled meiosis? Put your hand up. Yeah, you've already done this practice. That's what you were being assessed on. Okay, so not much to say here. If you understood meiosis earlier, it's the exact same things that we're talking about here. Maybe what I'll do then is it's a good opportunity to quiz you all again, crossing over random segregation, independent assortment. So let's start with Regina. What is crossing over and when does it happen? Which prophase? 1 or 2? Okay, prophase 1.
It's when the chromosomes swap the genes.
Any chromosome swaps genes? Remember, you have to be very specific. Good. Chromosomes, so adjacent chromatids of a homologous pair randomly exchange genetic info. You see that extra level of detail added there? That's the level you want to have for a bad success. Adjacent chromatids. Okay? So, for those of you who remember, this is a chromosome when it's replicated, right? That's one chromosome. That's the sister chromosome here. And we call the material that a chromosome is made out of, we call that chromatin. And since they're attached to one another, we call these two sister chromatids. Very good. So what we're saying here is adjacent chromatids of homologous chromosomes randomly exchange info. That's crossing over. Good. Abhishek, what about random segregation?
What happens in phase 1, and it's the movement of homologous pairs to either side of the cell. So if one homologous pair moves to the right, then the other has to move to the left.
Very good. Do we see that happening there? And have a look at what you're being shown here, right? So, give me another image. There we go. Yeah, have a look at this. This is modeling random segregation. Have a look at your notes, right? What you see is a scenario where the blue chromosome moved to the left and the red moved to the. But in the other scenario, so in the first scenario, the two blue moved together and the two red moved together as well. In the second scenario, what happened? They went to opposite directions, right? The red actually moved with the blue, and you see that happening on the other side as well. That is random segregation happening right in front of you. That is how you model random segregation, right? So a little pointer to all of you, if you ever asked in a trial paper to model random segregation, you need to show both possibility. You need to literally write arrow, possibility 1, arrow, possibility 2, right? Now, the reason why there are two different possibilities, even though the blue chromosome moved to the left in either one, is because it was inherited with a different chromosome in each of the possibilities. Do you all see that? We don't care whether blue moved to the right or to the left. We care what it was inherited with. Does that make sense? So what I mean by that is, this is one scenario. Everyone agree? But what if the blue moved to the right, and what if this blue moved to the right? Would that make any difference? No, because the two blues would be inherited together, and the gamete would have two blue chromosomes. That's why they're showing you a case with blue and blue and red. Those are the two possibilities. Okay? Question to all of you, is this modeling independent assortment? Very good, because we're looking at two different homologous pairs too. Random segregation is just looking at one, right? So we're saying if this chromosome randomly moves in this direction, the other must move in the opposite. The second you do that, you've modeled random segregation, right? Independent assortment is what we're modeling with as two possibilities. That's what we're truly modeling. So when we write possibility 1, possibility 2, as you can all see here, that's modeling independent assortment. Okay? Very good. Any questions so far? Yeah? Yes, do you see, both have to do with the movement of chromosomes. The chromosomes move in phase 1. But, again, there's no point. So when we talk about metaphase and anaphase, for a cell, it's all happening continuously, right? So that's why some textbooks would say it happens at metaphase 1. They're not wrong, because it lines up ready to move in metaphase 1. So it's just nitty-gritty detail differences, but both would get you the mark.
Excuse me, sir.
Yeah?
With this model, aren't the chromosomes supposed to be ready recombined, like crossed over already?
Yes, they shouldn't.
Yeah, okay.
It's a very good point. I like that you picked that up, because if this was a 3-mark question and you're assessing the model, that would be a huge con. They've not shown crossing over. It's a very good pickup. Good. Good. All right. Good job. All right, let's move on. Okay, now coming back to this, we'll quickly talk about alleles and the type of inheritance. Now we're going to actually talk about genotypes as well, which I haven't spoken about yet. Okay? Now, for every gene, how many alleles do we have? Typically, in reality, we have way more than 2. But for HSC Biology, we're going to reduce things as simple as possible. There are 2 variations of the gene. Now, question, what is a gene? That codes for? Not an amino acid, polypeptide. That is the definition of a gene. An amino acid sequencer of polypeptide. Good. Now, in HSC, you need to think of 2 variants of this gene. So if we're talking about a certain gene, let's say we're talking about the gene that codes for eye color. Okay? The alleles are variants of that gene. So I'm going to write next to alleles, variants. Now, again, in HSC, we're simplifying things. In reality, there's like 10 to hundreds of different alleles, even for one gene, and there's hundreds of genes for one trait, right? So things like height. Do you all see how humanity has a bell curve of height? A lot of people are in the middle. A few people are very short. People who are very tall, right? That tells you there's not just 2 different variants. Otherwise, everyone would either be very short or very tall. The HSC was simplifying things a lot, okay? So we imagine that we have 2 alleles for a gene. There is a dominant one and a recessive one. My question to all of you is, in what cases would the recessive allele be expressed? In what cases would the dominant allele be expressed? So when we talk about alleles, the way we denote them is a capital A would represent the dominant allele. You can use any letter you want, but the capital and the lowercase have to look different. So it can't be C. C looks the same upper and lowercase, right? So you need to pick something like A or R that looks different, right? So we'll pick A. So little a represents a recessive allele. So let's pick a person and let's talk about height, okay? If a person had, let's say the dominant trait for height was tall and the recessive trait was short. If an individual was capital A, what would their phenotype be? That's their genotype. That's the 2 alleles they have. That's their genotype. What would their phenotype or physical trait be? Tall. Very good, right? Because that's only the alleles they have. Very good. What about little a? Very good. They both have 2 recessive alleles, right? They're not being masked by anything. There's nothing dominant. So they'll both be expressed because they're not being masked in any way. This is going to be short. There's a phenotype. So this is genotype, this is phenotype. What about capital A, little a? Yeah, so what's happened here? Did you all see the dominant allele, which in this case was tall, has masked the recessive allele such that only the dominant allele makes the protein and codes for the trait. The recessive allele has pretty much been silenced completely. It does not contribute at all to the physical characteristics. Does that make sense? So we would say the phenotype here would be tall. Now, did you notice we defined the dominant allele based on this specific kind of genotype, right? The only way we know that tall is dominant is when an individual had both alleles, they expressed the capital A, which became dominant allele. So how would you define a dominant allele? Very good. Now, you've used a very interesting word. Heterozygous. Let's break that down. What does zygous mean? I need to make the connections. Where have you heard of the word zygote? There you go. So it's telling you it's about the fertilized cell from the spermatozoa egg, which is every human at one point in time. What does hetero mean? Yeah. So this is a heterozygous genotype, right? Because it's got 2 different genes. The zygote has 2 different alleles for that one gene. Does that make sense? So the allele that's expressed in a heterozygote is the dominant allele. That's how you define the dominant allele. Does that make sense? So how would you define a recessive allele? So you just say the inverse. It's the allele that's not expressed in a heterozygous individual, right? So that's how easy it is. Now let's talk about how we name these genotypes. If you have two dominant alleles, what do we call them? Good, homozygous dominant. And if we have capital A, little a, we said it's heterozygous. Do we say heterozygous dominant? It's a bit redundant, right? In heterozygous individuals, always a dominant allele that's expressed. So you never say heterozygous dominant. And then homozygous recessive. Good. On that note, we will finish today. And for tomorrow, next week, we will, what I'll get you all to have a read of. I'd like you to read up until page, I think the page numbers are different for you, but could you all go to the ABO compatibility? I want you to finish reading up until these questions. Okay. So what page is that for all of you?
Page 4.
Okay, yeah. The numbers are just a bit different, but it should be page 12 for that inquiry question. Yeah. Yeah, that's so everything up until ABO compatibility I want you to read up on. Very good. Good job. Any questions at all? Good. Anyone have any assessments that are coming up? Now, preface to all of you, those of you who have assessments, please make sure you give me at least 3-4 days to have a look at your work, okay? You can't send it to me the day before and expect a response. Very unlikely you'll get one, okay? So make sure that you do give that leeway in case that you have that, okay? And always, if you have an assessment task, tell me the date it's due so I know how urgent it is to get back to you, okay? Good. See you guys. Bye.